Hi, everyone. Is this thing on? Okay. Good evening. Welcome to the Norma L. and Roland G. Ware, Jr., Professor of Biology and Neuroscience inaugural lecture. I'm very pleased to be with you, all of you, here tonight on this occasion. And tonight we celebrate our newest Norma L. and Roland G. Ware, Jr., Professor of Biology and Neuroscience, Hadley Horch. And we recognize her extraordinary accomplishments and contributions both to our academic community and to her field. This is an important occasion for Professor Horch and her family, friends and colleagues and students, and a well-deserved honor. And it's a significant occasion for the college as well. It gives us the opportunity to highlight our belief in and support for the people who exemplify the kind of research, teaching, and learning that is at the core of what we do. Professor Horch embodies exactly what makes our faculty so special and what makes Bowdoin's model of education so powerful. She is both an influential scholar and an inspiring teacher. The professorship we are celebrating tonight was made possible by Roland Ware, who is a member of the Bowdoin class of 1954, and his wife, Norma. The Wares were modest and kind people who loved Bowdoin and disliked public attention. They quietly established their first chair in 2009. When Roland passed away in 2021 and Norma died a year later, they supplemented their original gift with a generous bequest that allowed for the creation of several new professorships. I'm so happy that Professor Horch is the second of 3 faculty members named to new Ware professorships. Professor Horch arrived at Bowdoin in 2002 after a year in postdoctoral research at Cornell. She received her undergraduate degree in biology from Swarthmore College and a PhD in neurobiology from Duke University. In addition to receiving research awards from the National Science Foundation and the IDEA Network of Biomedical Research Excellence and publishing numerous articles in neuroscience journals, she's co-editor of a 2017 book, The Cricket as a Model Organism. Also in 2017, Professor Horch was chosen by our students to deliver Bowdoin's Karofsky Encore faculty lecture. Hadley Horch is a pioneering researcher, a dedicated teacher, and a somewhat obsessive champion of a species of crickets, a particular species from the Mediterranean, commonly known as the two-spotted cricket. Her research seeks to understand how the cricket's molecular neurobiology and changes to those structures affects its regeneration, development, and behavior. Professor Horch is passionate about the cricket's unique set of strategies for solving neurobiological problems. In her lab, for example, she and her students have studied how crickets can regenerate and reconnect at a neuronal level in order to recover from injury to their auditory system. Professor Horch's lab is a place of significant neurobiological research. It's also a place, and I was learning a little bit more about this tonight, where students experience substantial intellectual and personal growth. Each year, a group of students flock to the Horch Lab to conduct research over the summer, research that frequently leads to original discoveries that become the basis of honors projects and sometimes co-authored publications in science journals. Hadley's thoughtful and committed mentorship, and her infectious enthusiasm for crickets inspire students to dive deeply into research year after year. The Horch Lab is an incubator for future researchers, creating a multiplying effect as Hadley's students carry forward the neurobiological knowledge and research skills that they have learned, as well as the passion they have developed for scientific discovery. Professor Horch's influence also extends widely through her field and community. Here's just one recent example. In 2018, Professor Horch received a major National Science Foundation grant for her research into the changes in neuronal plasticity in crickets as they age. The project, entitled Adult Compensatory Plasticity in an Invertebrate Sensory System, contributes to our understanding of the flexibility of older nervous systems of many different organisms, and gives us insight into why recovery from nervous system injury varies from individuals to individual. Based on this research, Professor Horch went on to co-author articles with several of her students in the prestigious journals BMC Genomics and the Journal of Comparative Neurology. Hadley and her students also shared their discoveries with local elementary schools in and around Brunswick and with Wabanaki middle school students as part of a coordinated program of STEM outreach. This is just one example of the ways in which Professor Horch exemplifies Bowdoin's teacher-scholar model. Her work advances our understanding of the resilience of the nervous system and provides the basis of further scientific and medical breakthroughs to aid recovery from nervous system trauma. At the same time, her inspiring teaching and nurturing mentorship help launch generations of her students into further scientific study and discovery. Put simply, her impact is profound here at Bowdoin and beyond. Beloved as a teacher, mentor, and colleague, and respected as a leading scholar in her field, Professor Horch is a deserving appointee to this chair and recipient of this honor. Please join me in congratulating and welcoming to the stage the Norma L. and Roland G. Ware Jr., Professor of Biology and Neuroscience, Hadley Horch.
Thank you, everyone. Can everyone hear me okay? Yeah. I'm a little overwhelmed. That was very kind. Thank you very much. It's really, really nice to look out in the audience and see so many friends. Some I haven't seen too recently, so thank you, everyone, for being here. I'm very honored. I'm very humbled. Like I said, I'm a little, it's really nice. Thank you. Okay, so I would like to tell you a little bit of a wide-ranging story today that's going to be in two separate parts. And they might seem a little disparate at the beginning, but I'm going to magically pull them all together at the end. So part one is going to be sort of how I got here, and part two is going to be where I am going. So we're going to start by thinking just for a second about gardeners. So when you think of gardeners and what gardeners do, you might think about establishing, planting things, fertilizing, nurturing, maybe pruning back or thinning, shaping the plants as they go. And what I'd like to tell you today is a story about a set of molecular gardeners, so proteins, that basically garden or shape neurons. So before I jump into that, I want to give just a little bit of cell biology background for maybe a few people who haven't had this in the top of their minds recently. So I'm encouraging you to think about a cell, imagine a cell as a factory. So factories produce all these things, and inside a factory, there's probably a central office that has all of the instructions for everything that factories make. So we can think about that as the nucleus, and the instructions for everything that they're going to make is the DNA, the chromosomes, the genes inside the nucleus. And there's lots of different kinds of factories, and not all factories make the same subset of output. So sometimes a factory just makes a portion of what is in the instruction manual. So think about that as a subset of instructions, maybe even a card that sort of has some sort of specialty instructions for making just one thing, and that's actually the messenger RNA. So this messenger RNA might make a green protein, not really, obviously. And so the factory output are these proteins, each of which come from these kind of cards based on the instruction manual. And within a factory, some of those proteins might be for the factory. They might be used very locally, or they might be exported. They might be released from the cell to actually out into the wild, the spaces between the cells. They might actually be studied on the membranes of those cells so that they can interact with proteins from other cells. So, whoops, wrong button. So here is another factory, another cellular factory, and it might make a protein that can act as a receptor for the protein made by this cell. So in this way, cells communicate with each other. So neurons, which I'm going to talk about today, are just specialized cells. This kind of factory where the instruction manual is called the cell body. It's right here in the center. And most of what you see here are dendrites. We often refer to them as the dendritic arbor because they look an awful lot like branches of trees. And dendrites receive the information from other neurons, and then this cell will sort of decide what it hears or what it senses, what it collects. And from here, it will send that information down its axon, this next process that kind of comes out of the cell, so that it can talk to the next cell and the next cell. And through all of this, the information kind of moves through the circuits of the brain. So the gardeners that we're talking about today have a specific name. They're called neurotrophins. So a little bit of a goofy name, but neurons, obviously, for neuro. And trophins comes from troph in Greek, which means nourishment. So you can really think of them almost as fertilizer. So with a broad brush, the things that neurotrophins do is they support the survival of neurons, and they modulate neuronal connectivity. So those connections between dendrites and axons are actually influenced by neurotrophins. Okay, so part one is just a little bit of a historical journey. I don't often talk as much about history, but I wanted to tell you about the people who discovered these, and it's really kind of an astounding story. So neurotrophins were discovered by Rita Levi-Montalcini, who was working in the lab of Victor Hamburger. And I want to tell you a little bit about Rita's life. So here she is in 1909, she was born in 1909, this is obviously a little bit later, in Turin, in Italy. And here she is at 97. She lived to be 103, and she won the Nobel Prize for her discovery of neurotrophins. So she went to medical school despite the protestations of her father, who did not think that was a great idea for her. And she joined Giuseppe Levi's lab and worked with luminaries and other Nobel laureates such as Renato Del Becco and Salvador Luria, names that my biology colleagues will recognize. So in 1938, when she was working in the lab, they passed the Defense of the Race laws. So because of her Jewish ancestry, which was evident in her surname, she was not allowed to work anymore. So she fled Italy at that time, or left Italy to go to Belgium, where she worked in a lab there. And there she learned the chick embryonic system. So you can open up an egg and see a teeny, teeny, tiny embryo. And this was a very important system early on that was used to study the development of organisms. So she was there for only about a year until 1939 when Hitler invades Poland and she feels she has to return to Turin. So she moves to her family, she moves into her childhood bedroom and decides she's going to set up a lab. So she buys herself a microscope, she gets slides, she goes to farmers and asks for eggs, fertilized eggs, and she uses sewing needles to do the little microsurgeries that are done on these embryos in her childhood bedroom. So Mussolini and Hitler declare war on France and Great Britain. Great Britain starts bombing Italy, of course, and especially Turin, so hundreds of planes would sometimes go overhead. So when the air raid sirens would go off, she would pack up her bedroom lab, her microscope, her slides, carry it all downstairs into the shelters, wait out the raid, and bring it all upstairs again, night after night, year after year. So just to give you a sense of what her life was like in this period, this is a picture taken on her street in Turin of a destroyed house just down the road. So she wrote in a letter during this period that she was amazed at, quote, the complete possibility of escaping to the present, diving into the marvelous charms of her work. Her niece at one point noted that Levi-Montalcini kept working to survive. So in 1940, she is reading Victor Hamburger's work, And he was been working on these chick embryos, and he was doing a pretty simple experiment where you'd go in and you remove this little purple tissue here, which is the nascent limb bud, the tissue that's going to grow into the leg of the chicken. And so the question was, the cells that are born in the nervous system, in the spinal cord right here, if they don't have a target to grow into, what happens to them? And so when Victor Hamburger did this experiment, he removed the limb bud and looked about 5 days later, and this is what he saw. So on the intact side, where the limb bud was there, you saw a nice healthy population of neurons. And again, these are the motor neurons that are going to grow in, innervate that limb, and talk to the muscles there. But on the side where the limb bud was missing, you get a much reduced population of neurons. And so he posited that there must be some factor in the limb bud that actually induces the cells in the spinal cord to become neurons. And when that factor is gone, very few of them turn into neurons. So Rita Levi-Montalcini in her bedroom laboratory said, okay, I'm going to do this same experiment. And instead of just looking at day 5, I'm going to look at day 2 and day 3 and day 7 and 8 and up to day 19. She was going to do a play-by-play every day, extremely careful, extremely thorough experiment. And what she found when she looked really early was something a little surprising. So on the intact side and the side where she had removed this little limb bud, I know this little red dot is a little hard to see. Hopefully you can see this. The little limb bud, the population of motor neurons was equivalent between the two. There was no reduction in the side where the limb bud had removed. And in fact, this population of cells was quite robust. There were a lot of them there. And as she looked over time, she started to notice this hallmark of some unusual things happening inside the cells, and they were dying. So the cells that were on this side, if they didn't have a target to connect with, they would die. So what she discovered is that neurons are overproduced before they grow into the limb buds, and that the cells that grow into the limb buds, they compete for survival factors. So there's something in the limb buds that they need to survive, and the neurons are competing for those. So in 1942, her publication options were limited. She was not allowed to publish in Italian, the normal journals. So she found a French journal in Belgium, and she published some of this work. And she managed to find a Vatican journal that she could actually publish in Italian, a little bit of a loophole somehow. And this is just an example of some of the work that she was doing, again, in her bedroom laboratory. She was drawing and taking photographs. And this paper in particular, there are 45 pages of detail, including 7 pages of photographs and drawings. And I also want to just give a shout out. I learned about a lot of this kind of somewhat hidden history by this really wonderful piece by Bob Goldstein that was published in the Nautilus in 2021, where he goes back to Turin, sort of walks her pathway there, follows in her footsteps, and finds that French paper and that Italian paper and interprets them, and comes to realize that this thing that we completely know about the nervous system, which is that neurons are overproduced, and the beautiful sort of specificity of connections that gets formed happens because of what she observed, which is that you need that fertilizer from the target. And so this finding has been attributed to multiple other people over time, but it belongs to her, and nobody really knew it because of the war and everything else. Incidentally, just a little bit more history here. So Victor Hamburger was also impacted by the Nazis. So he grew up in Germany. He worked with Spemann in Freiburg. And he came to Chicago in 1932 on a Rockefeller fellowship. And when he was there, he received a letter from the chancellor of Freiburg, who happened to be Martin Heidegger, the philosopher, discharging him from the faculty because of his Jewish heritage. So Rockefeller had established a fund for displaced German scholars, and they kept them working and employed, and eventually he ended up at Washington University in St. Louis. So it seemed like a timely time. It seemed like a good time to include this story. I find it really inspirational. And for me, it highlights the resilience and curiosity inherent in the human spirit. It also highlights something I don't think anyone here needs, reminding of: the devastating impact of discriminatory rules. They stymie forward progress. They lead to brain drain. They don't don't help. And it also just reminds all of us that we can turn to something that we love. It doesn't have to be science in times of stress and danger, and that projects we're passionate about can really bring us some calm and center us. And I just, again, find it very, very inspiring. So around 1942 or after the war, Victor Hamburger was reading Levi-Montalcini's experiments, and by all accounts was delighted that she had come to a different conclusion than he had, and invited her to come and work in his lab. So together, they decided, like, okay, we know this is happening, so what is the fertilizer, right? What is this magic serum that it's getting from the limb buds, and can we find out what it is? So these are the experiments that Levi-Montalcini did that ended up winning her the Nobel Prize. So she took a clump of cells out of an animal, and she grew them in culture. So you'll have to trust me that this is a clump of cells. And you can see some little processes kind of coming out. So these are neurons that, they're doing okay. They're kind of hanging out. They send out a few processes, nothing too exciting there. But if she took that limb bud and sort of ground it up and added that to the culture, this is what she saw. Oh, that's good. So she saw this halo of new neurite growth, so neurons growing processes out. And so that's great. It's obviously coming from the limb bud, but there are many, many things in the limb bud, so what is it? And so now her job was to winnow and winnow and winnow, narrow down what she was adding to this little test to see how many fewer things can she add and still get this kind of output. And this story is completely off the rails bonkers, and I don't have time to tell all of it, but it involves tumors and snake venom and male mouse salivary glands, not female mouse salivary glands, that all helped her narrow down, work that she did along with Stanley Cohen as well. They shared the Nobel Prize for this. And eventually ended up adding just one thing, one protein, and which they called nerve growth factor. And when they add that alone, they get this beautiful halo of growth. So since that time, this was in the, I think by the 60s, by the time that this was all done, we have learned a lot of things. So first of all, this is nerve growth factor. This is what it looks like in its kind of ribbon protein form. And groups after it started to look for other factors that might be similar. So one example is using the exact same readout test. Some people went into the brain and said, all right, is there anything in the brain that actually will do this as well? And they discovered another factor called brain-derived neurotrophic factor, because it was derived from the brain. And that you can see what that looks like in purple. It looks pretty similar to what the NGF looks like. And then as time marches on and we get more and more data, eventually we have databases where we have lots of protein information. And now people can dig through proteins. So up here, what you can see is the sequence for NGF in the amino acid sequence, and BDNF. And any place that there's a star, it means that it's very similar or identical to the amino acid in a third neurotrophin called neurotrophin 3. They got increasingly inventive with the names. So this is neurotrophin 3, again, found at the protein level by looking at sequence similarities. So if I kind of pull up a little cartoon, this cartoon of the shape, I want you to kind of imprint on this shape, keep an eye on it, maybe remember it. There's a couple things that kind of almost like a little T shape. And then down here in the base, I don't know how well you can see this, there's some C's that are connected with a line. So these are cysteine bonds that are binding to each other. And so this is what is called a cysteine knot. So this is a very stabilizing structure that helps this protein take its shape. And so this is what you find in all neurotrophins, is that they all have cysteine knots. Okay, so what do we know so far? We know that neurotrophins are proteins that are released by cells, that neurons compete for those neurotrophins, and that when they get them, they are nourished, right? They get nourishment, they survive, they send out axons, their axons grow. There are multiple neurotrophins, NGF, BDNF, NT3, and and that they have a conserved shape. They have this cysteine knot. I didn't really tell you this, but we now know that they're found in all mammals. So they're a very well-conserved protein that's found kind of across the across the mammalian set of animals anyway. So I want to remind people of a little bit more neuroscience stuff. So here is our neuron that we were looking at, the dendritic arbor that receives the information and the axons that pass that along. And it was clear even 100 years ago that the axons from one cell would grow towards the dendrites of another cell. And that's when we kind of started to figure out that these are probably talking to each other. And in fact, they are. So axons come in like this, and they make a connection with a little gap space connection to a dendrite, and this is called a synapse. So this is where axons and dendrites talk to each other. And I should also point out at this point that the activity that is flowing down these neurons and these axons is basically electricity. So action potentials are a form of electricity that flows down the axons. That signal then jumps the synapse, and then the dendrite receives that, and the electrical signal sort of continues from there. Another thing that I want to point out is that dendrites, and neurons in general, but if we think about dendritic arbors, they come in all sorts of different shapes. And I'm making a slightly different point here. Hold on. First of all, I want to point out that the reason that we can see this neuron is that we have, well, we didn't do it, Cajal did it, but filled it with a stain called Golgi stain that made everything turn black. And if you imagine this to be about 1 millimeter across and a little bit more than 1 millimeter tall in a cube, there would be 57,000 neurons in that chunk, right? So it's extremely dense. And so if everybody was filled with, this black dye, you wouldn't be able to see anything at all. So one of the kind of things that's tricky for neuroscientists is that we need to be able to look at neurons in isolation if we want to tell what they look like. So an example of this, these days, we use a lot of fluorescent tracers, fluorescent dyes, to be able to fill one cell so that we can see. Even though there's thousands of cells right in this region, we can see the shape of one. And then finally, the other thing I want to point out here is that the shape of these dendritic arbors is radically different between cells. So the cell on the left probably receives about 10,000 synapses, and the cell on the right probably receives 200,000 synapses, right? So very, very different shape and size and very different function. So there's a really strong connection between form and function here. And you might also imagine, like, whatever the rules are, whoever's sort of in charge of growing these dendrites and making them look like they do, if all the dendrites were over here on the left of the cell rather than the right, that's totally going to impact who they can talk to, right? What circuit they're going to be part of. So how you shape these is really fundamentally important for how the circuits of the brain develop, and then how we all function, what our behaviors are, all that kind of stuff. So we know our molecular gardeners influence survival and axonal growth, but do they also influence dendritic growth? So that was the question that I asked when I joined the CATS lab at Duke University for my graduate work. So here's just a small example of that work. This is a cell from the brain of an animal, which I filled with something fluorescent, so you can see a really nice little dendritic arbor there, fairly simple. And what I did in another set of tissue is that I made the cell fluorescent, but I also cranked up the level of BDNF. So I kind of went into that cellular factory and said, let's make more BDNF protein. And when you do that, that's what these cells look like. So you can see it almost is just a little bit like that halo, Rita Levi-Montalcini's halo. There's lots more dendrites than you see normally. And if we look over time, we know that control dendrites are really very stable. Maybe they grow a little bit, but there's not a big difference 24 hours later. But if we look at these cells over time, here's an example of a dendrite that's here that wasn't even there at 26 hours, and dendrites that were there then disappear. And so if I quantified this, when I quantified this over a number of cells, if you look at the hatched bars here, they represent the cells with the cranked up BDNF. And so dendrites are lost and gained at much higher proportion than they are in the control cells, which are shown in the solid bars, which almost never lose dendrites and slowly sort of gain them. So, okay, I told you about these things, and now I can add a little bit based on my work and, of course, the work on many other people. So neurotrophins also influence dendritic development and growth. And then, just because it's interesting, I'll summarize many, many decades of work. Neurotrophins are very involved in mood disorders. So we think that BDNF levels are really low in people who have depression, especially depression that's hard to treat. Exercise increases the levels of BDNF. And we also know that it plays a role in injury. So in spinal cord injury, BDNF has a local role to play, and we also think a therapeutic role to play. Some people are thinking, working on adding it to see if we can help neurons recover from injury. Just another example of basic research, basic curiosity question turning into something that absolutely has health benefits for us. All right, so that wraps up the history part. And it puts me right at the end of my graduate career. And I was in a little bit of a pickle because I had gotten the job at Bowdoin, which was amazing, but I was working in a really expensive vertebrate model system that wasn't appropriate to bring to Bowdoin. So luckily, Bowdoin gave me a year to go do a postdoc. So I kind of hunted around for a really interesting dendritic question in an invertebrate, in an insect of some kind. And I came across the work of Ron Hoy. So Ron Hoy was at Cornell. And so I'm going to tell you a little bit about this insect model system. Ron Hoy had basically spent his career doing a lot of different things, but was very interested in auditory communication between bugs. And in fact, he spent much of his career just finding ears on bugs because there are a lot of bugs we don't know where the ears are. So they're all over the place. They're on the side, they're on their head, they're everywhere. So let's talk a little bit about the cricket and about the auditory system of the cricket. So this is Gryllus bimaculatus, the Mediterranean cricket, which is the model system that I work on. And males chirp, just like all crickets do, by rubbing their wings together. That's that delightful sound you hear on summer evenings. And female crickets find that super duper sexy. So when a male is chirping, a female will hear that and say, okay, I hear that sound coming from the right. I'm going to walk, hop, jump, fly, whatever I need to do to get to this male so that we can mate. But the other thing that crickets pay attention to is bat ultrasound. This is not a sexy sound. This is a scary sound. So hunting bats make these bat ultrasonic chirps as they are hunting for insects at night. And crickets and moths and a number of different insects can hear that, and they will move away from that sound because they do not want to get eaten. So if we zoom in on the auditory system, we can find the ears, which are actually out here on their forelegs. Now you are all in the know. Little tympanal membranes on the front and the back of their legs. And if you zoom up on their hairy little legs, this is what they look like. So on the rear, on the back side, there's a very large, it's basically just an eardrum. It's a membrane that vibrates. And on the front, there's a tiny little one. And so sound comes through, vibrates those membranes. The axons underneath, that turns into an electrical signal there. And those action potentials zip up the axons here, and they go into the central nervous system, into this green blob. So it's called the prothoracic ganglion. It doesn't matter. I might just refer to it as the PTG. But if you want to just think of it as a ball of cells, that's basically what it is. That's fine. And that's where the auditory system is received into the central nervous system. And we'll go inside that in just a little bit. So what might be a little bit obvious with ears way out here is that they are vulnerable, right? If you lose a leg, you've lost an ear. And crickets lose legs easily. So they molt 7 or 8 times before they become an adult, and sometimes they don't get out of their molt well and they lose a leg. And if they've lost that front leg, then they're deaf on that side. They also are under predation pressures from, bats, but also frogs. So if a frog tongue happens to get this leg, the cricket will actually drop the leg. So they have these things, they're called autoptic tendons, where they can just drop the leg and hop away and be safe. And it's good to give up the legs. They don't really need it, and they survive. But of course, that means that they are deaf on that side as well. So Ron Hoy was thinking in the mid-'80s, like, I wonder what happens to this whole auditory system when you lose an ear, you lose a limb and then an ear. So if we dive into that green blob, that PTG, this is part of what's going on inside there. So these 2 gray claws coming in from the left and the right represent all the axons coming from the ear. And you can see it's this bilateral system, left to right. And they make synapses onto the next order of cells. So we can look at the one on the right in black. And those are all dendrites. So those are all dendrites receiving the information from the ear. And then this one single process, it's really hard to see, the axon is going up to the brain. So the cell receives the information and sends that up to the brain. And then the brain decides, did I hear cricket or did I hear bat? And was it on the right or was it on the left? And then makes a corresponding behavioral decision. So if we cut off the leg, we cut off those cellular factories, and all those axons die. So the axons themselves here in this claw are going to disappear, and it's going to leave this black cell disconnected. It doesn't have anyone talking to it for the moment. And so it could die, it could hang out, it could do a number of things. But what this cell does is it sprouts across the midline. So this is the center of the body. Usually respect that, but this one sprouts across. And it actually grows right towards the axons coming from the opposite side, and it gets functionally connected to the axons from the opposite ear. So it's deaf for a couple of days, but as soon as it grows across, it's basically recovered its sense of hearing again. And I can tell you the short answer is we don't know how they do it, and we don't know why they do it. So we don't really understand what advantage it gives you, because the sound is still coming in one side. It's now just going up to both sides of the brain, and it's not clear why they do it. There's a lifetime of work to be done. So let's look at a slightly more simplified version here. So in yellow are the axons coming from the ear. Green is a stylized version of the cell. If we cut off the leg, we lose the axons. Our cell is disconnected. And now it sprouts across the midline, which typically serves a little bit as a barrier. It grows and it branches, and it forms new synapses with the axons on the opposite side. So I can tell you in 2001, whenever I was looking at this stuff, it looked a lot like a job for neurotrophins to me, right? There's growth and branching of dendrites, synapse formation, which I didn't tell you is something else that these neurotrophins can do. So I was super excited that I'd be able to kind of ask a very similar question in a different model. Unfortunately for me, in the early 2000s, everyone was pretty sure that insects did not have neurotrophins. So if we go back to this protein alignment here, and this is comically silly, but if you tried to look through a fruit fly database, a Drosophila database, you found nothing. You didn't find anything that was convincing that it was a similar protein in fruit flies. And the field had sort of decided they're not very flexible, they're hardwired, they don't need neurotrophins. That's just, that's not what they do. So, okay, that's fine. We had some other things to chase. So I brought the model system to Bowdoin. And what we spent several years doing was basically applying more up-to-date molecular techniques to this system. So down in the corner is Ron Hoy's backfill. This is what they did. And incidentally, this probably exactly was done by Steve Hopman's wife, Jane Hopman, who they were working at Cornell in the mid-1980s, and a lot of the technical work was done by Jane at that time. So we managed to figure out how to backfill, how to put dye, fluorescent dye, into these cells. And with our fancy confocal microscope, we can get a three-dimensional image. And now we can do some really nice measurements of volume and surface area and all sorts of stuff, kind of get into the nitty-gritty about what these cells are shaped like. But the question of why this was happening and what was, the cause was sort of what we wanted to tackle. So instead of guessing, okay, it's neurotrophins, because maybe it wasn't, we decided to do an unbiased screen. We decided to ask, all right, what's changing? And we did a number of these, and in the early days, they weren't very sophisticated because we didn't have a lot of tools, and it's just gotten better and better over the years. So this is the most recent one that we've done. And this was work done by Harrison Fisher, who's now, I believe, in biomedical engineering at BU, super talented student. So he took one set of crickets and he cut off the leg, which makes the ear go away. And so now within that PTG in the center there in white, it's half connected, half not, right? And because we wanted to control for the stress and everything else, we took another set of crickets and we just cut off the very tippy end of the foot. So trauma and stress and whatever whatever this animal was sort of responding was hopefully going to be controlled. So he can then go into these animals and he can dissect out that PTG 1 day after he's cut off the ear, 3 days, 7 days, and so on and so forth. And he can do the same in the foot chop ones, the ones without the foot. And so then what we do with that tissue is basically send it off to a company and help us do a lot of this because this is expensive and difficult. But what we wanted to do was assess the changes in gene expression. So we wanted to look at all of these instruction cards that are present in those cells and ask, are the same instruction cards seen in this animal at this time versus this animal at this time, or did they really change? So just give you a quick example of that. Let's imagine a cell in our animal where we've just cut off the foot. You've got a couple instruction cards for a green protein and a whole bunch of instruction cards for an orange protein. But if you look at a cell in the animal where we've cut off the ear, maybe you see something like this, where there's many more green cards. And so that means that whatever the instruction for the green proteins have been upregulated. There's more of them there, you're gonna make more green proteins. And similarly, or oppositely, maybe there were a lot of orange proteins here, but you make a lot fewer after you cut off the ear, and those are downregulated proteins. So we can go through and look at all of these and try to understand what things are upregulated, what things are downregulated, and that might give us a sense of what's going on here. So you get a lot of data with these. A shout out to Felicia Wong, who I think is a newly minted MD at this point, and she did the first pass of data here. And what we have here is what's called a volcano plot. So if we look at this axis down at the bottom, the x-axis, on the right are changes in level that have gone up. So those are our upregulated genes, and on the left are our downregulated genes. And then on this axis is whether it's statistically, like the strength of the statistical significance. So mostly you want to focus on these red dots, because those are the ones where we have pretty big, strong effects. And as you can see, there's many dozens. In fact, there's hundreds of genes that are up or downregulated. So you can comb through lists, and it gets a little bit overwhelming. But a more recent tool, and something that's more helpful that Joel Graber at MDIBL is helping me with, is a functional analysis. So this is where you can sort of look at all of those proteins, and you can say, wow, I can see that these 10 proteins are actually doing something similar. They're in the same pathway, the same network. Let's look at those levels. And if that group is up and down regulated in some sort of interesting way, that might hint that that pathway is actually really involved and really important. So we did that functional analysis. We found quite a few things, but one thing jumped out at us, which is the toll signaling pathway. And now you may be thinking, you're done, your brain is full, no more, but just one more step here. We're almost there. So the toll signaling pathway looks like this. It starts with a protein called Spätzle, and yes, it's named after a German noodle, and it binds to a receptor called a toll receptor. Two of them actually kind of give it a little hug. And when the toll receives the Spätzle, the postsynaptic cell, or sorry, the cell here at the bottom realizes that there's been some communication to it from the outside. And we know that the toll signaling pathway is very important for very early development, like insect development. That's been known for a long time. That's actually where the Spätzle name came from. Spätzle. Thanks. And the other thing that it's involved in is the immune system. So those were two things that we knew it did. And there might be an immune function here, but that wasn't super interesting to me. But the more I dug into this, the more I found this. This is what Spätzles look like. So maybe this looks familiar to you. If you followed my directions and remembered things from the beginning, you get this sort of T shape, and look down here. 1, 2, 3 cysteine bonds. You have a nice little cysteine knot at the bottom. So if we compare these to each other, NGF and Spätzle look very much alike. So structurally, they're very, very similar, even though the receptors are a little bit different. And so the question is, functionally, are they similar or not? So this is where my two disparate parts of my story come beautifully together. Hopefully you agree. So let's just look at the red BDNF here. I've told you that it's involved in cell survival, it's involved in axon outgrowth, it's involved in dendritic growth, and my little asterisk is there for my graduate work that contributed to that. And if we look at the Spätzle function through toll receptors, and this is all work done fairly recently within the last 10 or 15 years from Alicia Hidalgo's lab working in fruit flies in Drosophila, they have found that it's very important for cell survival and axon outgrowth, which leaves dendritic growth. And we seem to be pretty well poised to ask this question, right? In an invertebrate model system where the Spätzles and the tolls appear to be regulated upon the loss of an ear. So I don't have answers for you today, but I'm going to just walk you briefly through the experiment that we've tried and the one little piece of evidence right now that looks very promising. So if we take an intact animal, we can experimentally decrease the level of Spätzle or toll. And we're going to focus on toll 6. There's many different tolls and many different Spätzles. And if we do that, when we do that, we're going to then measure the impact on function and the impact on the anatomy. We're going to look at these dendrites and sort of see. And the way that we can do this is that, remember, these axons grow all the way up to the brain, And if you cut open a little piece of the head capsule, the brain is right there, and you can lower an electrode down onto the brain, and if you play sound to the animal, you can actually listen into their responses, right? You can see the action potentials all the way up there as they're responding to, say, bat ultrasound. And my colleague, Bertolt Hedwig, just figured out this tremendous thing, which is if you can put dye in this electrode, you can push it through into the brain, and it will go all the way back down, and it will fill the dendrites of that cell. So we've got function and anatomy all together in those cells. And I shout out to Sam Brilwell, who came with me and Patsy Dickinson to England to learn this technique, and he successfully brought it back during the height of the pandemic and got this working in my lab, which I'm forever grateful for. So very quickly, the predictions from this is that these animals are going to have an altered ability to respond to sound. So when we listen in on these action potentials, it's going to be a little different. We think that the dendritic morphology is going to be altered compared to normal. And if we were actually to test the behavioral responses, its ability to respond to sound, those would be different as well. So I'll give you this one piece of evidence. This is beautiful work done by Jada Scotland as she was completing her honors thesis. Pretty sure she's med school bound this next year. So she could put that electrode on the brain, give sound stimulus, and then these are the responses she'll see, like little action potentials. We call them spikes sometimes. And she'll do this in control animals and animals into which we've lowered 6-1, one of these tolls. So first she can look at the latency, and she can see that in control animals, they respond more quickly than in animals that have had decreased 6-1. She finds, if she looks at burst duration, how long they fire for, that these cells in animals where we've reduced 6-1 fire less long. And if we just count spike numbers here, we also see a decrease in spike number. So there seems to be a functional response to lowering the 6-1. So I cannot tell you right now about the morphology. Stay tuned, that's coming. These experiments are a little tricky to do, and we've had some snafus, but we are gonna be able to backfill these cells and be able to get morphological data. And the next kind of frontier for us is behavior. So if you take a cricket and suspend it in a wind stream, they think they're flying. They don't always flap their wings, but they'll tuck in their little legs and they'll basically respond to sound. So what you'll see is the abdomen will be jerking to the right. There was one right there, and there, and there. So we're playing bat ultrasound from the left, and the cricket is hearing it and deciding that it is gonna go to the right. So we've got a couple talented students working on this. Ryan's in the audience right now. I don't know if Tom's here too. We're collaborating with Jack O'Brien in math, who's helping us with the analysis of this as well. Okay, I realized, so first of all, I'm slightly over time, I apologize, but I also realized I probably could have spent a half an hour of this whole talk just thanking people, because there are a lot of people to thank, but I'll go through quickly. So first of all, of course, Norma and Roland Ware for the gift to the college. Really appreciate, obviously, the gift. I'm very honored, and to the family, it was an incredibly generous gift. I wanted to thank my family, many of whom are here tonight, actually. I've got one kid in Germany who didn't make it, but almost everyone else made it. So thank you for being here. And thanks especially to Fred, my husband. He is my biggest cheerleader, and I appreciate having him in my corner. I wanted to thank Kathy Sawicki at Swarthmore College, who taught me everything about neuroscience from the beginning and really made it very exciting. I did a 2-year technician job with Peter Sargent at UCSF, who is just a superbly careful scientist and kind of got me going on the graduate school route. My graduate work was with Larry Katz at Duke University. We lost Larry tragically very young. He was a phenomenally insightful and creative scientist, and I really appreciated my time working with him. And then the absolutely delightful Ron Hoy, who let me come to a postdoc for 1 year and have a baby and learn everything about crickets. He was really great. I wanted to thank, in particular, many people at Bowdoin, but Patsy Dickinson, who could not be here tonight, but she was just such an incredible mentor from day one, helping me with both teaching and research. I really very much, I don't know what I would have done without her. And to all my biology and neuroscience faculty and all the folks in Druck, thank you. You guys answered a lot of really dumb questions from me early on as I was moving into a new area, and I appreciate it. And this room is amazing, full of friends that when we met at the Children's Center, right, when our kids were really little. So I really appreciate everyone being here and for your generosity and friendship over the years. I have 3 fantastic technicians to thank. So when I first started, when I got my first grant, Sarah McCarthy was my absolutely fearless technician who just jumped in and figured it out, and I don't know where we would be without her. I got to work with Justin Beckman for 1 year, who was great. And then most importantly, Lisa Ledwidge, who's here somewhere. Everybody knows Lisa is spectacular. Lisa keeps the wheels on the bus. She is smart, she is a deep thinker, she helps me figure stuff out, she's always calm, she's always there. I've been working with her for almost a decade now, and I'm not sure what I would do without her as well, so thanks to Lisa. Lots of funders to thank. This was mentioned at the beginning, but I've had funding directly from NIH and via Maine INBRE, NSF most recently. A shout out to Corey Colwell and Cindy Stocks in the Office for Student Fellowships and Research, who help us facilitate this kind of tremendous amount of work with students every summer. The Paller family and Neuroscience for their fellowships. And then, importantly here, I've already introduced you to Harrison Fisher and Jada Scotland and Sam Brillwell and Felicia. And I wanted to point out that there are 152 additional polar bears who have worked in my labs for at least a semester or a summer or something else. And so I wanted to thank all of them, and I was going to give a special shout out to those who are in my lab this year. So Janiyah Bradley, Elijah Dumdy, Kyla Gary, Tom Hahn, Sammy McLemore, Ryan Kong, and Ronald Tran. So thank you very much. Thank you. We do have a little time for questions, and I've been told that if anyone who has a question should come to the microphone. You can certainly ask anything that you want.
I have 2. Did you hand place every polar bear there?
I actually did.
That's, oh,
It seemed like overwhelming. It actually wasn't so bad, but. And there's only 4 or 5 actual different kinds of polar bears there. But
yeah, I noticed that. I was like, how did you... Anyway, so I don't know if you can flip slides really quick, but
Yep.
I was curious to see the SEM photo of the cricket's leg with the ears.
Yep.
Again. Yeah, wow, that was quick. Are those on, because I was thinking about binauralism. That's how you can tell which direction a sound is
Exactly.
coming. Could they use could the mini ear and the big ear on one leg
Yep.
compose a rudimentary binauralism?
Yeah.
Would that explain the regrowth of dendrites towards one leg?
I don't know that it explains that, but it does explain crickets are, one-eared crickets, one-eared females are actually kind of remarkably good at finding males, and that it shouldn't be that way because they only have one ear. And actually, so a normal female will walk in this weaving pattern, and basically they have this really simple algorithm. If it's louder on the left, now it's louder on the right,
Just like
and
the
so they
bats.
just do that. But when one-eared females circle, like this. And I've often wondered, so that big eardrum on the back and that smaller eardrum on the front, they might be able to detect whether the sound is coming from the front or the back.
Yeah.
And I suspect that's probably why that behavior happens. But it was that behavior that influenced or inspired Ron Hoy to do that experiment in the first place, and a number of other things. So why they grow over and why it's important to have sound going up both sides, even though it's only coming from the intact side, is just a total mystery. So the folks in my lab who are working on the behavior, we're basically flying these crickets. We can do it in intact animals, and then 3 days after we cut off the leg, 7 days after we cut off the leg, as this system matures, and the hope is that we'll be able to tell some subtle things about they'll be able to tell us what they're hearing, basically, is the hope.
Very cool.
Yeah, thank you.
Thank you.
Here.
Super cool talk, Hadley. Thank you very much. I learned a lot. That was great. That was kind of my question. But now that I'm obviously here, I'm going to ask my secondary question. I was fascinated by the fact that these crickets, their neurons go to a place that's not their brain. Is that a typical thing for insects, or does it teach us anything about hearing in insects versus any other animals?
So it's not actually all that unusual for nervous systems. So there are often these relay stations. So even your hearing comes in and ends up in your brainstem first, and then in your thalamus, and then in your cortex, where it's the first time you're sort of perceiving that sound. And like in the spinal cord, there's lots of local circuitry. In fact, that local circuitry can actually help you walk, right? There's the circuitry for walking is there. It's just controlled top down. So it's not actually that unusual. What's unusual is that, the cell bodies are way out on the outside, which is weird. They really do seem to be literally just little protein factories that kind of pump proteins into the dendrites and the axon. So why, in those other pictures I showed you, the cell body was right smack dab in the center of everything, and for whatever reason in inverts, they grow differently. They're different. So the short answer is, no, it's not that unusual to have a relay station. And so that information gets shaped and sharpened here, and then when it gets passed up to the brain, there's another level of kind of shaping and sharpening. Okay. So the nervous system often works like that.
If there's no question right behind me, let me just follow up on the hearing part. Are there people who are working on maybe equivalent, like if a person goes deaf in one ear, do we know if they can sort of, I don't know what I'm asking, I guess. What are there people who look at deafness in people that way?
So I think one of the reasons that this model is so incredibly compelling is because it's so unusual. So most of the time, especially in adult nervous systems, if you disconnect a neuron like this, it's not going to sprout and all of a sudden recover its function. And so that's one of the reasons I really wanted to study this, is because we might learn some fundamental rules about how nervous systems are able to recover from injury that we might be able to apply to other systems. And I'll give just one more little smidge of information. So the cell that I've shown here, it responds to bat ultrasound. There's another cell right on top of it that I didn't draw in here that responds best to crickets chirping. So there's this kind of bilateral system going up there. And when these grow across into the other side, this one maintains its bat ultrasoundness. So it's finding the proper partners to remain responsive only to that ultrasound, and the other cell that's responsive most to crickets is finding other synaptic partners. So there's this really beautiful specificity that happens between these cells where they're finding somehow the right partner. And again, how they know how to do that is anybody's guess. And I've got hundreds of genes to work on for the rest of my life, so some of those might be doing that.
Thank you.
Thank you.
Thank you. That was so awesome. And I've seen this picture so many times, and I've thought new thoughts after this talk. That's so exciting. So the gray claw is multiple axons. It's a bundle.
70.
Okay, about 70. So what I'm wondering is, with the dendrites of those cells, and thinking about the two membranes, front and back, is there anything known about whether they're different cells that might be taking the information from those two membranes, and then is that connected with different dendrites synapsing
Great
or something
question.
like that? Seems impossible to figure out. But.
So there is, underneath those two membranes, there is a tympanal organ, and that is equivalent to our kind of inner ear, I guess. And nobody really knows anything about them. It's incredibly delicate. It's very small. It's out on a tiny little cricket leg. And I think when I first, as I got into this lab, I was like, I'm going to figure all this out. And it's just really, really hard. So no one even knows if there are dendrites out there. There's probably some sort of stretch receptor cells or something that's vibratorily responding to the sound, and that's how you actually get these electrical action potentials that then pass along to the next level. But as far as I know, there's almost nothing known about the tympanal organ. Like the actual ear of the cricket is very unclear. There's been a ton of work done, relatively speaking, on this central system because it so beautifully shapes the sound. And they know it's louder on the right, but this system here locally makes it even louder. So it amplifies the differences. And that's one of the reasons the brain can confidently say, yep, this is coming from the right, and I don't like it, or I do like it.
Hi. In one of the slides, you illustrated your control of just cutting off the cricket's little toe compared to the experiment of cutting off its whole arm, ear situation. And earlier, you also mentioned that crickets have some mechanism by which they can release their arm if it's just taken away.
Yes.
Is that ever been something you've measured, the difference between the voluntary release versus a traumatic sort of loss of limb in the dendritic growth?
That's a great question, and the answer is no, we've never done that. It is, I mean, the effect at the end of the day should be the same, because when you rip, if the leg gets released, basically the nerves are being severed as well. They're severed slightly a little bit higher than we sever them, but it's not all that different. Ron Hoy did this really beautiful experiment at one point where instead of severing the leg, they actually did a little tiny surgery. They went in and found the nerve coming from the ear, and they crushed it. And so by crushing it, you lose the you lose the axons going to the ear. So it looks just like this on the right, but then because the whole nerve is intact, those axons regenerate and they grow back in, and then they can reinnervate that black cell that's just grown across. And then the question is, what the heck does it do then? And the answer is still quite bizarre and something I'd love to look at. That cell maintains input from both sides, which is a really kind of not supposed to do. They're supposed to choose one or the other. So it's now connected to both sides. So behaviorally, what does that do? So yeah, so that didn't really, I answered a question I wanted to answer instead of that. But yeah, I don't know the answer to that. And there might be differences in like other, hormone or stress processes with an autoptic loss versus a severing. So it's a good question, but I don't know.
Thank you.
Yeah.
And just one more general question. Do you have any personal speculation on maybe the evolutionary advantage of having ears on your arm? I'm curious about that.
No, and I don't know a ton about bug ears. And all I can tell you is that they really are everywhere. On grasshoppers, they're right here on the side of the abdomen. They're sometimes out at the ends of the antenna. They're on the legs on all cricket species. I would not be able to speculate on that. It's a good question, and there's probably people who have speculated on that, but I'm not one of them.
I'm curious, what happens if you do sort of the in-between manipulation? So don't destroy the whole leg, the nerve, but just one of the ears, the large or the small.
Those tympanal membranes?
Yeah, so do you get like a partial reach over, or it seems like that could be a little confusing.
Yeah, that's a great question. We've never done that. The tympanal organ is right there. It is really hard to destroy the membrane without destroying the tympanal organ itself. So that experiment of Ron's is somewhat along those lines. The experiment we'd really love to do, which we've tried, which is also very hard, is that instead of cutting off the leg to remove all those axons, what if we just silence the axons? Because then you've got contact, you've got pre and post synaptic, because they've got sticky outy proteins that are talking to each other. So there's communication happening there that has nothing to do with the electricity. So if you could block the electricity but keep the contact, would this happen or would nothing happen? So we actually tried, TTX is the toxin that some animals make that paralyze, they block the channels that let the action potentials go. So we tried to make teeny tiny little blocks of Silgard with TTX in it and implant them locally so that it would just locally block the axon. But they're so small and they're so sensitive, the animals would just die. So we really could not ever we just couldn't fine tune that enough to make that happen. We tried cooling. We also thought about cooling it, because cooling will block those action potentials. If you keep them cold for 3 days, could we do that? We literally tried to hand feed them and little droppers of sugar water while they were cooled. That didn't work either, but it's a great idea. Do you want to ask? We're almost out of time, but.
No, my question was about what happens when they grow their legs back again, but you answered that already.
Well, did I?
I thought you did. I thought you were saying that was what Ron Hoy's experiment showed, that they stay innervated on both sides.
Well, that was just the axons. So if you remove the ear, the legs will regenerate, but the ear never regenerates. So even if you take, little baby crickets are like the size of an ant. If you cut off the leg then, they will regenerate a very normal looking leg, no ear. Tympanal membrane and everything, no ear. That's another mystery. So many mysteries.
This is going to be a malformed question, but I'll try and make it quick. like consider the entire population of crickets as an organism. Losing a leg could be a symbol that like maybe that one shouldn't reproduce. So yeah, if being able to direction.
Is there a fitness question here?
Yeah, like maybe sometimes you wouldn't actually want that dendritic regrowth to happen if the cricket was like a bad cricket and got bitten by a frog or something.
So I won't answer that question, but I will answer a question that I want to answer again. It turns out that when we look at this growth across the midline between males and females, it's different. There's a sexual dimorphism here. So in these pictures, we either measured what we called the maximum perpendicular extent, how many dendrites, how far the dendrites grew across. So these are just the dendrites growing across the midline, or we traced like the longest one. And the data was insane. It was all over the place. And what we found, my wonderful student, Alex Pfister, who wrote everything down in her lab notebook, she had males and females, yes. So, and I did this work with Amy Johnson and Olaf Ehlers. They helped me curve fit this. So the males are in blue, really nice, beautiful, this is over time. So we looked at 3 days and 5 days, kind of like Rita Levi-Montalcini did, and saw that we have this really linear growth curve. But in females, they zip across really fast, and then they plateau. And by 20 days, the male's dendrites are actually twice as far. They've grown over the midline twice as far as the females. We don't know why. We've never, all of my, all the genomic or the kind of genetic data that we've got is all males only because of this linear curve, but someday we'd like to do males versus females. And our only guess is that, males build a little burrow, chirp, send out that sound, and females are out jumping and hopping to get to the male, so they may be under greater predation pressures on their way to get to a male. So maybe there's some impetus for them to get there fast, and then it doesn't matter what happens after. I don't know if there's an advantage or a disadvantage to growing your dendrites twice as long. It's another behavior experiment that we could do. Ryan's gonna have to stick around for multiple years and finish a bunch of stuff in my lab. Esther wants a question.
First off, hello everyone.
Hello, Esther.
Dr. Horch was my neurobio professor. Quick shout out to your boots. They're very cute.
Thank you.
However, my question is kind of dumb, but I'm very curious. Have you so the audience knows I'm a yapper, so I don't have complete sentences. But have you measured action potential, amplitude, and how that changes, in your control versus the experimental group, and is there a difference?
So no, we have not measured that. So because action potentials are all or none, within one animal we wouldn't expect there to be difference. Oh, it's so tempting to ask you a question right now, but that
oh, no.
axon diameter is probably going to influence the size of that action potential, right? So yes.
Science.
So there may be some differences there, maybe, that is an interesting question. We have never done that. And I'll also say we are doing extracellular recordings, which do not give us that information. If we were doing intracellular recordings, we'd be able to do that, but those are super, super hard, so we don't do that.
Understood. Thank you.
Thank you. I think we're well over time, so I bet they want us to stop now, but thank you very much.